Our next awardee for engineering, and the presenter is Dr. Michael Deem, who is John W. Cox Professor of Bioengineering and Professor in the Department of Physics and Astronomy at Rice University. Now, if you didn't hear me, let me repeat. He is Professor of Bioengineering and Professor in the Department of Physics and Astronomy. He wants to cover all of it, so nothing is left out. He was recipient of NSF prestigious Faculty Early Career Development Award, along with many others. He's a Fellow of American Institute for Medical and Bioengineering, American Physical Society, and American Association for Advancement. Please welcome Dr. Deem.
Well, thank you for the introduction. I'd like to thank the Academy for this award. Looking at the other awardees this year and the awardees from previous years, I have to say the Academy is really doing a great job of promoting science, technology, engineering, and medicine in Texas. Personally, I'd like to thank Rebecca Richards-Cordham for nominating me, David Helms for all his support at Rice, and the awards committee for their work. It's a real honor to represent Texas in this way. So what I'd like to talk to you about today is some work that I've been doing over the last 5 or 6 years in the search for fundamental mathematical laws in biology. And this title will make more sense as I sort of describe that program. So this work has been done by a number of the people in the group. Zhang Kui, he is a postdoc now at Stanford. He's soon to take up a position in China as a professor. Jung-Man Park is a longtime collaborator. He's a professor of physics in Korea. K.L. Pan's now a postdoc at MIT. Jun Sun works for a geotechnical service company now in Houston. Ramdas Pofle works for a financial service company in Houston. And Enrique Munoz is a professor back in Chile. So I'll give you a brief overview of this, what we call FundBio program of DARPA. I'll then describe my interest in the formation of structure in biology. I'll say a little bit about this equation PE equals M prime over R. And then I'll turn to immune system dynamics. I'll talk about vaccine design and vaccine effectiveness. I'll talk about dengue fever and influenza. And for those of you who haven't seen it, this is one of our newest buildings at Rice called the BRC. The 3 National Academy of Science members that we just hired, they'll get the top floor here, and my office is over here somewhere on the 7th floor. So there was a program in DARPA to search for fundamental mathematical laws in biology. And it's sort of surprising that there should be such a program, but it was really a pleasure to be part of it. And it was really started at the behest of the director of DARPA at the time, Tony Tether. And the way the story is told is that he got a bunch of program managers together and he said, you know, I get these engineering and physics proposals and I can sort of evaluate them, right? I have physics and mathematical laws. If we get a proposal that seems to violate the second law of thermodynamics, probably we're not going to fund it. But I get these proposals in biology and I don't really know how to evaluate them. I don't seem to have any mathematical principles to guide me. So, you know, aren't there some fundamental mathematical principles in biology? And I think maybe there was sort of a subtext here that there are a lot of very smart mathematicians who work with DARPA, and I think maybe Tony Tether wanted to get some of those mathematicians working on some of the more modern aspects of biology. After all, Dr. Tether was an applied mathematician himself, so I think he wanted maybe to support the field a bit. So at Rice, our chairman of mathematics at the time was Robin Foreman, shown here, and he had one of the planning grants to get this program off the ground. So he recommended me to DARPA. And Ben Mann and Eric Eisenstadt were the 2 program managers who really took up the challenge from Tony Tether. This is Ben Mann. He lives in Austin now. So those of you involved with these large government programs know there were 3 planning meetings, right, building up to the program. Most of the biologists gave up in meetings 1 or 2. Ben Mann is a pure mathematician, so he kind of selected against the physicists. So it ended up being basically 10 mathematicians and 10 biologists who were part of the program. The proposal went up to Tether, and he accepted it. So it was really great. I mean, it was pretty nicely supported over 5 or 6 years, and it was a pretty good group of people that made up the team. So 2 of the biologists were elected to the NAS during the program, and 2 of the mathematicians were as well. And the elevator pitch for the program from Ben Mann was that we were to find F equals ma for biology. So we wanted to find something like Newton's laws, maybe a set of laws, that each of which could look something like Newton's laws for biology. So phase 1 was to propose some mathematical laws. Phase 2 was to solve some of these mathematical laws and then to design some experiments to test them. And then phase 3 was to test some of the solved laws. And then there was a continuation year, which was a little bit more applied. So what I'll talk about is the main law that emerged in this program, and it was something that we developed at Rice. And it's related to this P equals m prime over r. And the idea is, how does structure arise in biology, and what are the forces that promote this development of structure? So I'll sort of circle around this idea for a few slides. I want to say a little bit about modularity. In biology, we see modularity at multiple scales. So here's a protein. Proteins are made up of atoms. Atoms make up amino acids. Amino acids make up secondary structures. So here's an alpha helix. Secondary structures form domains. A typical human protein has 3 to 5 domains. This is actually a multi-protein complex. So there are really 14 proteins here in this enzyme. So modularity seems pretty prevalent in biology, and why is that? So this picture is sort of nice. So Physics Today put it on their cover of my article in January, and then they reused it in the December for their holiday card. I guess it sort of looks like a wreath. Not only are proteins modular, but genes are modular, right? So you have 23 pairs of chromosomes. Within your chromosomes, you have your genes, and then within each gene, you have introns and exons. So there is this hierarchical organization of structure, and genes or proteins seem to evolve by concatenation and reorganization of this material. So here's an example taken from the human genome sequencing effort. We start with this EPC-like protein. Adding the Ph.D. domain gives this. Adding the BR domain gives this. And then adding the zinc finger and the BMB domain gives peregrine. And that's not an isolated example. Here's what I showed you. Here are a few other examples of expansion of human chromatin proteins. And here are some more examples of expansion of other regulatory proteins. The last setting I'll describe modularity in for you is in networks. So here is an example of the developmental pathway of the sea urchin. This is from Eric Davidson, a pretty well-known developmental biologist at Caltech. And always when one looks at these types of networks, there are subnetworks that seem to have structure. Uri Alon has looked at this in a quantitative way. So he looks for statistically overrepresented subnetworks in a big network. This is from the transcriptional interactions in E. coli. And when he identifies these statistically overrepresented subnetworks, he calls them motifs. He makes a dictionary of them. So for example, here's a feed-forward network. So I would call these modules. And this is what I want to focus on, is how do we sort of quantify modularity, and then how do we describe why modularity exists. So biology is a subset of chemistry. I would say it's the modular subset or the hierarchical subset. And the emergence of biology is really the emergence of this modularity. It's a symmetry-breaking phase transition. Modularity is the order parameter for this phase transition. So the general result that I want to talk about here in the first bit of my talk is under these 3 conditions, modularity spontaneously emerges. So if we have an evolving system in an environment that's changing, the fitness landscape upon which the evolution occurs is rugged. So evolution is in some sense slow. And we have horizontal gene transfer. So essentially, if there's genetic exchange of information between different individuals, under those 3 conditions, then modularity will spontaneously develop. You'll spontaneously get this biological-like structure out of all chemical possibilities. And if we had to write down an equation, it would look something sort of like this. PE equals m prime over r. M prime is the rate of growth of modularity. PE is the amount of environmental pressure. And r is telling us how rugged the landscape is. So the essential idea is the more changing the environment, the more the system needs to adapt. And the modules that the system spontaneously develops help the system to be adaptable. And we see this in engineering all the time, right? We make everything out of units because that allows us to both to diagnose the system, but then also to respond to changing market forces. So I want to give you half a dozen examples of this law in biology. So the first one is from Ursulon. What he did was look at about 300 different bacteria in different physical environments. And he ranked the environments in terms of their variability. So maybe tide pools, for example, would be quite a variable physical environment. He then calculated the modularity of the metabolic networks in these different bacteria, and lo and behold, we see this monotonic increase of modularity with environmental variability. So exactly what we would predict. Here's an example from our group. What we did was look at the protein-protein interaction in yeast, and we looked at the protein-protein interaction network at current time, but also at older and older evolutionary times. So going forward, here is the protein-protein interaction network from a couple billion years ago, and here is running evolutionary time. The modularity increases with evolutionary time. It sort of saturates. So I first presented this in front of a general DARPA audience, and one of the cardiologists said, I don't know, that's a little weird because usually we think evolution is accelerating, but this is saturating. So that kind of spurred us to calculate a second order modularity. So we looked at the modularity of the module-module interactions, and that's shown here. I've lined up the axes. So right when the first order modularity is saturating, the second order modularity is taking off. So in general, biology is developing really a hierarchy. So it's developing modularity at multiple scales. And this is what allows biology to kind of span the space of evolutionary possibilities. We see modularity in physiology as well. So these are some data from a group at Duke, from Tim Buckman. They're analyzed by Gunnar Carlson, who's a pretty well-known topologist at Stanford. He looked at healthy subjects, congestive heart failure patients, and atrial fibrillation patients. And he's looking at the heartbeat signals processed in a certain way at long, intermediate, and short times. And at all times, healthy subjects have two modules, right, sort of an upper and a lower in this space. Congestive heart failure patients have two modules at long times, but only one at short times. And atrial fibrillation patients really just have one module at all times. So modularity is a sensitive diagnostic of health. Health has these natural fluctuations. These natural fluctuations are in this modular form, and it allows the physiology to adapt to changing and stressful environmental conditions. And the younger you are, and the more healthy you are, the more fit you are, the more modularity there is in your physiology. Here's another example. This is looking at functional magnetic resonance imaging data from brains. So basically looking at metabolic activity in the brain as a function of position and time. And the modularity of young brains is greater than that of aged brains. Here's an example from another group. They looked at the modularity of control subjects and epileptic patients. And again, control subjects have more modularity than the epileptic patients. Here's an example from ecology. So we looked at streams in New Zealand, and the idea here was we want to look at small streams where a lot of the energy input to the stream is from detritus from the banks. So we looked at the amount of organic matter in those streams, and the idea is that if there's very little organic matter, there's very little energy, it's kind of a very stressful environment for that ecology, there should be a great amount of modularity. If there's a lot of particulate organic matter, it's not very stressful, there's a lot of energy, there's no need to be as modular. And that's exactly what we see. So the modularity decreases as the stress level decreases, as the energy goes up. Another example I'll come back to in the second part of my talk is for influenza. There's one protein, hemagglutinin, on the surface of influenza that your immune system recognizes. And really the immune system recognizes one of 5 modules on that protein. And we can watch the evolution of influenza really by looking at the composition of those modules. So here is influenza over the last 40 years. Evolution kind of proceeds along this curve as depicted in this 2-dimensional space. So you have the quasi-species of the strains of influenza that infect us. It stays as a module, but its identity changes over time. So I'll come back to that one. The last one I want to relate to you is of a social network. So here I picked the World Trade Network as an example. So the network is imports and exports between country I and country J. And the United Nations has very good data on this import-export phenomenon for the last 40 years or so. And we can calculate the modularity of that World Trade Network. So what we would expect is the modularity should be going down as globalization has proceeded, right? Friedman told us the world is flat. So that's what we see, the flattening of the world. The globalization leads to less and less modularity in the World Trade Network. So that was already known. It was good to confirm that. We made 3 predictions. One is that recessions are like a change of environment. So I've shown here 3 major recessions and 4 minor recessions. And the modularity should go up during or after those recessions because that's sort of an induced change of environment. And we see that in 6 of the last 7 recessions, the modularity goes up a little bit. Another prediction is that the World Trade Network should be more sensitive to recessions now than it had been in the past because there's less structure now than there was in the past. And that structure allowed the system to be more adaptable. And that's what we see. The World Trade Network is more sensitive to recessions now than in the past. And then the last prediction is that the World Trade Network should recover more slowly from recessions now than it had in the past because there is less of this modular structure that would allow it to respond more quickly to changes. And that's exactly what we see. The recovery rate is slower now than it was in the past because there's less structure now than there was in the past. And these, at least all 3 of these predictions, especially the last 2, were sort of novel to some economists. So it was kind of satisfying to make these predictions from a fairly general theory and then see that they agree with the data, but also they're kind of new in a field in which we're not experts. So let me now turn to the immune system, and I'll talk about dengue fever and I'll talk about influenza. Dengue fever is a fairly serious viral disease as transmitted by mosquitoes. About 2.5 billion people live in the 100 or so countries in which dengue is endemic. There's a very small amount of dengue in Texas. We screen mosquitoes for it in Texas. And about 50 to 100 million people get infected with dengue every year, and about 500,000 get a more serious case of dengue, which used to be called dengue hemorrhagic fever. What's interesting to me is there are 4 serotypes of dengue, and the immune system recognizes the different serotypes to varying degrees. So here are summarized experimental data from human clinical trials in Thailand done by one of our collaborators, and what's shown is the response of the immune system, so the ability of the immune system to kill cells infected with the 4 different serotypes of dengue after having received a 4-component dengue vaccine. And this vaccine is not good enough. So what this graph is saying is, okay, you recognize these serotypes pretty well, but you really don't recognize these serotypes well enough. Shown in open is a spin glass theory that we developed of the immune response to dengue. And I'll come back to that spin glass theory when I talk about influenza. To summarize, we're very satisfied that it agrees so well with the data, but really this theory gave us an idea, which is the following. The immunodominance is competition for access to antigen. The T cells are trying to get access to the 4 different serotypes with the associated antigens. And we select for T cells, right? So there has to be competition in the T cells. This competition occurs in the lymph nodes, and humans have hundreds of lymph nodes, basically around all of your joints. So what if you vaccinated with serotype 1 here, serotype 2 here, serotype 3 here, and 4 there? Then you might make good T cells against serotype 1 here, 2 here, 3 here, and 4 there. And theoretically, that's a really good idea. It almost eliminates immunodominance. And there are now several proof of principle experiments. So HIV is a disease similar to dengue in which it's somewhat controlled by T cells and also in which immunodominance is somewhat of a problem. So there were a couple experiments where they showed if you did this multi-site vaccination, you got a broader T cell response than if you vaccinated in a single place in mice. There was an experiment in monkeys with dengue, which also showed if you did this multi-site vaccination, you got a broader T cell response. And then finally, there's a Sanofi patent where they cite this multi-site vaccination idea in the description of their dengue vaccine. So it's kind of nice that this spin glass theory eventually led to something that looks reasonably practical. Now, this multi-site vaccination also works for cancer. That's actually where we first read about the phenomenon of multi-site vaccination. Hans Schreiber is an oncologist. He used to be at MD Anderson. He's now at the University of Chicago. And he did experiments where he showed if you did single-site vaccination, so if you put 2 different cancer vaccines in the same place, you got a response against one of the components much more dominantly than the other. But if you did multi-site vaccination, you could get a more nearly uniform response to the two different components. So we can explain those experimental data with this theory that I briefly described, and then using that understanding, we were then able to generalize it to vaccines and apply it to dengue. So now I want to conclude with talking just a little bit about influenza. So influenza comes in various strains. I'll focus on H3N2 because that's what we have most often got over the past few years, and it's the most serious form. So we hear a lot about H1N1, and that was very serious in 1918, but because we've been exposed to it so long, our immune system actually recognizes H1N1 pretty well. And H1N1 vaccines are even better than H3N2 vaccines. So turning that around, H3N2 is actually a little bit worse for us than H1N1. Here are shown the efficacies of the H3N2 vaccine that we get over the last 40 or so years. And they're shown as a function of this order parameter P epitope that we developed. P epitope came out of a spin glass theory for the immune system. And sort of skipping over all the theory, the main point is that the efficacy of the vaccine in humans actually correlates pretty well with this pretty simple order parameter. So it's something that we can calculate just from sequence data. And many strains of the flu are sequenced every year. So this is an eminently calculable quantity. The correlation of vaccine efficacy with P epitope, it's about 0.81, the R squared. The correlation with measures of antigenic distance you can get out of ferret animal models is about 0.4 or 0.5. So this is what I think is probably a pretty rare case where the theory actually works better than the animal models for this prediction of the vaccine efficacy. So we're kind of happy with that, and we've been talking with flu people for the past 5 or so years about this. And let me conclude with this point. There's a great interest in predicting what new strains of virus will come about. And the influenza is a good model system for this. So the flu virus evolves every year, and typically one of the components in the flu vaccine is changed every year because of evolution of that flu virus. And to give you a sense of the timescales, we need to pick in February what strains will go into the flu vaccine for the vaccine that people in the northern hemisphere will get in October or November. So we need to predict half year to a year in advance what will likely be the strains that we'll encounter as a population. That's very difficult to do a priori prediction because there's so many directions in sequence space in which the flu can evolve, it's hard to say which one will be taken. Essentially, the entropic factor is really high. However, we can do detection. So that's what I'm showing you here. Here are data from 2009, and these are the sequences in a two-dimensional space that most of the people who were infected with H3N2 had. But there's this little cluster over here that is growing and is sufficiently far from the cluster of the current dominant strain, as measured by P epitope, that the immune system thinks it's different. And with those two criteria, we can state that this is probably going to be the dominant strain next year. So that idea works pretty well. Here it's a few months later, and you can see, yes, that cluster is growing. So this idea doesn't have any false positives, and it is a little bit more predictive than what the World Health Organization currently does. So it is possible to detect in the sequences that are currently collected the minority which will likely be the dominant strain next year by doing this projection and then by looking for clusters that are growing. So again, that's something that we're talking with people who know more about the flu to kind of convince them that maybe there's some useful information here. So to conclude, I talked about this search for fundamental mathematical laws, and I described the one that we developed at Rice, and this really became the dominant theme of the DARPA program. So it was a great program for us, great program for Rice. And if we have to write down an equation, we write something like this, that the rate of growth of modularity is proportional to the amount of environmental change, and the proportionality factor is related to how rugged the landscape is, how difficult the evolution is. This emergence of modularity really characterizes biology as distinct or as a subset from chemistry, and it's the order parameter of this symmetry-breaking phase transition. So modularity, I showed you examples of it arising in molecular biology, physiology, even social networks. And then I talked a little bit about this spin glass theory of the immune response. I described the idea of the multi-site vaccination. I described a couple of proof of principle experiments for HIV and for dengue. And then I told you a little bit about influenza. How can we predict the effectiveness of a vaccine? So if you're vaccinated with one strain of the flu, and then you see a different strain, how effective will that vaccine be? And then finally, the strains of the flu, they will evolve from year to year. So can we detect that evolution in its sort of, in its genesis, in its early phases? So with that, thank you for your attention. Thanks to the Academy for supporting everyone in Texas, and I'll be happy to answer any questions that you have.
Question? I'll ask you an odd question. If you convert me into modularity and then digitize it somehow, can you beam me to Washington tomorrow? 20 years from now?
I'm not sure about that one, but modularity does allow systems to adapt, right? So in engineering, we're really familiar with the concept of modularity, and sort of quantifying it in quantifying modularity in biology was novel. So we could explain different aspects of biology. Not yet the beaming you, though.
We can't get there yet. Any questions?
Right, so this is a phase transition. So the question is, okay, if I'm forced to write down an equation, I write down this Taylor series. We lost our equations. If I'm forced to write down an equation, I write down PE equals M prime over R, which is kind of a linear expansion around the phase transition point. The emergence of modularity, it is a phase transition, it's spontaneous. So certainly there is a lot of nonlinear physics going into that phase transition. This order parameter expresses the degree to which the phase transition has proceeded. So it's sort of like temperature, if it's above 0, then we have water rather than ice. So T minus 0 is some linear looking thing, and it might be a function of other things like pressure and so on raised to some power. So here we get this linear form near the phase transition. As the transition proceeded, if we looked at, say, the modularity and the protein-protein interaction, eventually it bends over, and we start to see those nonlinear effects. And when the nonlinear effects become very strong, that's actually when we see the second-order modularity taking off. So we're interested in right when the phase transition occurs, and then if we're interested in the hierarchy, so if we're interested in the multiple levels, then we tend to calculate this thing that I didn't really describe, but the CCC. That's the amount of hierarchy in the system. So we project the distance metric of the data onto the tree metric that best fits the data, and the CCC is the amount of the original data that you can represent by the best fitting tree metric.
So I made this maybe an oversimplification of what you're saying. So if you say you had, let's take 5 different bacterial strains, and one of them has, and there's different levels of differentiation to a particular environment. The most differentiated one and most adapted one to that environment would be less able to adapt. Is that the theory?
Right. So if the specialization is too great in any one niche, it will be difficult for that organism to adapt to new niches. So modularity is a way that organisms try to adapt to an environment, but not over-adapt. So the most optimal organism won't be modular, but if there's a requirement for that organism to be able to evolve to new environments, it will be somewhat modular. And the more changing the environment, the more modular the organism.
And so that follows to higher eukaryotes, where would it follow the idea of a stem cell to a differentiated cell and its ability?
Yeah, so I feel there should be something here that should tell us something about cancer, and we haven't really figured that out yet, but that's something I want to think about for the future.
Last question.
Thanks. So we know that the propensity to mutate is itself under selection.
Yes.
Is the propensity to become modular itself under selection?
Yes. So really, one of the titles I can use for this talk is Life Has Evolved to Evolve, or that the landscape upon which evolution occurs itself also evolves. So the amount of modularity can be under selection. So for example, the amount of regulation. We see more regulation in more complex organisms. That's a sense of higher levels of modularity. Also, the amount of this information transfer, the horizontal gene transfer that is related to R, it can be under selection. So we can see in different organisms, the higher the rate of horizontal gene transfer exists in organisms that are in more changing environments, at least in some subsets of pathogens.
I'm under the impression that biology will use the same molecule for a number of different functions. I'm not this is not my field, but is this the idea that the same molecule might be used over here for one part, in one network, and over in a completely different network, in performing a somewhat different function?
Right, so the question is, maybe paraphrasing a little bit, that there is, proteins can be multifunctional, and the one question about that is, were early proteins more multifunctional, or are more modern proteins more multifunctional? So I would generally say that early proteins were more multifunctional, and as the environment became more complex, proteins needed to specialize. And this is what people see both in experimental protein evolution and when they try to reconstruct phylogenies. So one of our, not the definition I showed you, but one of our definitions of modularity is how specific proteins are in their interactions. So if proteins interact with each other sort of through many domains, that's less specific than if they interact through just one domain. And the early proteins will tend to be more promiscuous, and the more modern proteins will tend to be more discrete in their interactions. So in a sense, that's a growth of modularity. Modern proteins need to be more discrete, more modular in their interactions, because otherwise they would have too many unintended side effects.
We'll allow one more question, last one.
In looking at this transformation from chemical behavior to biological behavior, you had 3 criteria early on. If I look at self-organizing systems, or if I think about early evolution, where you did not necessarily have DNA, what would be the equivalent of the exchange of information?
So it could be that, so the information has to be encoded somewhere. Either the information is encoded in the molecules, or the information is encoded in the networks that those modules form. And if it were encoded in the molecules, then it could be that molecules were spliced with other molecules. If it were encoded in the networks, then it would be exchange of parts of the networks.